Hi everyone, today I will present you about the HIV silencing and cell survival signature in infected T cell reservoirs. This paper is published in 2023 on the Nature journal. First, I will let you know about the HIV silencing in the infected CD4 T cells. HIV can persist in the body even during the antiretroviral therapy or ART treatment by hiding in the small numbers of the memory CD4 T cells known as a latent reservoir. These infected cells keep the virus silent, avoiding the detection and the destruction. Although the researchers have tried to reverse this latency with a drug called latency reversing agents or LRAS, the clinical trial haven't shown a clear reduction in the reservoir size. It means HIV still keeps silence in the CD4 T cells. A major challenge is that these rare infected cells are hard to find and study directly without altering them. Attempts to find them in the specific cell type have shown that the infected cells are spread out and highly vary. So identifying the unique features of these HIV infected cells remains a key hurdle in the developing a treatment. For studying about this, the single-cell technology played an important role in this study. Normally, the single-cell sequencing study have to extract the barcode sequence to each cell separately for easy identification. The barcoding method can be divided into two parts, including the barcode bead uses and the combinatorial indexing. The first one is the microbead, attaches with the barcode sequence on the surface. It uses barcode encapsulation in the droplet or ultra compartment with a single cells for attaching the barcode sequence to the genomic DNA or mRNA for itself. The second is the combinatorial indexing. This method is the barcode attachment inside of the single cells. Cells are fixed and permeabilized, followed by adding the barcode sequence in each cells and ligations and reverse transcriptions for the transcriptomic sequencing. From all single-cell sequence method, all cells are barcoded and sequences at once. It means CD4 T cells having silenced HIV DNA and naive CD4 T cells are sequences at once. For sequencing only the CD4 T cells having HIV DNA and eliminate the barcoding procedure, they develop and use the fine sequence of focuses interrogations of cells by nucleic acid detection and sequencing. Instead of co-encapsulation with a single cell in the droplet with the barcode bead, they encapsulate single cell inside bead. When talking about this technique in detail, first, for transcriptomic sequencing, accredited primer is attached with aryl agarose via aryl group and the inductions of the APS and TEMMET. Then this modified polymer is used for encapsulating single cells with a lysate buffer by using the microfluidic device. Then cells are lysed inside agarose beads and the mRNA inside bead will be released and attaches with the poly-T on the surface of the agarose bead, followed by the reverse transcriptions for converting mRNA to cDNA. Then for identify the bead, only having the target or HIV DNA, beads are reinjected with the Tagman PCR master mix and then droplet or digital droplet PCR. The droplet or bead having the target will express the fluorescent and isolated via flag equipment and stored as a single cell conditions. Then the only single cells having the target will be run sequencing. When focused on this work, the CD4 T cells were corrected and encapsulated by agarose polymer modified the poly-T oligo on the surface. Then cell was lysed and then released the mRNA. The released mRNA will be attached with the poly-T oligo followed by the converting to the cDNA by reverse transcriptase. Then all beads were reinjected in the Tagman PCR master mix with the GAG gene sequence that is specific genes of the HIV via the microfluidic device. Then the beads having HIV DNA will be isolated from the empty one by fluorescence detections and collected in the 96-well pair for further sequencing. This approach isolates whole transcriptome from cell containing quiescent virus without the need for in vitro latency reversal, thereby capturing a transcriptome-wide profile of these cells in their natural state. Next, they tried to use this method for studying the CD4 T cells having the HIV DNA. So they mixed the HIV DNA positive cells with the naive cells at 100%, 0.1%, and 0%, followed by the running 5 sequence. The results show that using the 5 sequence droplet cytometry can detect the rare HIV DNA positive cell with around 50% sensitivity and extremely low false positive rate, about 1 in 30,000. It means using this 5 sequence can separate CD4 T cells having HIV potentially. When 5 sequence was tested on the sample of the human and mouse cells, more than 99% of the genes expression data from the sorted HIV infected cells droplet came from the human cell. This shows that 5 sequence can accurately find and isolate the rare HIV-infected cells. Additionally, the difference in gene expression between two node cell lines using the standard method were very similar to the difference seen between HIV-infected cells and HIV-uninfected cells using the 5 sequence, further confirming the method's reliability. Next, they apply this method with the magnetically purified memory CD4 T cell samples from the people with HIV receiving long-term ART treatment that was initiated during the chronic infection. They compared the overall gene expression between HIV-infected cells and HIV-uninfected memory CD4 T cells. They found that the HIV-infected cells tend to group separately from the uninfected cells based on their gene expression pattern. Statistically, the difference between HIV-infected cells and HIV-uninfected cells were significantly larger than the difference among just HIV-uninfected cells. However, the sample tend to group by individual participants, and that the HIV-infected cells themselves vary quite a lot between participants. This means that while HIV-infected cells show the distinct gene expression pattern, there is also a lot of the variation between infected cells for the different people. To find out which the specific genes and the pathway were unique to HIV-infected memory CD4 T cells, the differential gene expressions or DGE was analyzed. When all participants were treated as the biological replicate in the combined analysis, they found 2,776 genes that were significantly different between HIV-infected cells and HIV-uninfected cells. Many of these genes were linked to cancer and cell cycle pathway, suggesting the difference in how infected cells grow and survive. Moreover, they found that only 11 genes that were differentially expressed in all 3 participants, showing that there are a lot of the variation between people. However, when they analyzed the effect of the biological pathway instead of the individual genes, 6 pathways were found that were consistently suppressed in the HIV-infected cells across all participants. This includes the pathway related to the programmed cell death and the pathway that normally limit cell growth. This suppression was due to both lower expressions of genes that activate this pathway and the higher expressions of genes that inhibit them. Even though the specific genes vary between participants, the overall pattern was clear, that HIV-infected cells tend to resist the signal that would normally lead to cell death or stop their growth, which may help them persist during the ART treatment. Next, the researcher used a method called WTCNA to group genes that behave similarly in the memory CD4 T cells with and without HIV DNA. They found 2 small groups of genes, including module 5 and module 28, that stood out in HIV-infected cells. These genes groups were linked to how genes are turned on or turned off, especially in the way that control RNA splicing, processing, and degradation. In Module 5, some genes were related to blocking HIV gene expression after transcription, which helped keep the virus silent. These genes were also involved in helping cells survive, avoid death, and continue to dividing, while the module 20X includes genes that help with the mRNA processing and protecting cells from dying. When they look closer, 8 of the 44 key genes were already known to affect HIV transcription, some of them blocked or others promoted. In HIV DNA positive cells, the blocker were more active and the promoter were less active. This showed the infected cells are not very friendly to the HIV activity. They also found that many of the other genes help process the RNA, protect the cells from dying, or supported energy production and repair system. Altogether, these findings suggest that HIV-infected memory CD4 T cells stay alive and avoid immune attack by keeping the virus silent and activated the survival pathway. Lastly, is the discussion part that I will more concentrated on the 5' sequence. The first, in this study, the researcher used 5' sequence to sequence the HIV DNA positive cells transcriptome. They found that the post-transcriptional HIV silencing and enhanced cell survival and proliferation is associated with how CD4 T cells cannot be eliminated after treatment. However, they couldn't distinguish between the intact and the defective HIV genome. These limitations mean that the further advances in 5' sequence are needed to get a clear picture. The second, the researcher points out that to get more accurate results from the 5' sequence, the technology needs the further improvement. This improvement would allow the better distributions of the data and more precise results across the different cells. And the last, with better technologies and longer study using the 5' sequence, they believe a new gene signature that were not detected in this study might be found, providing the deeper insight into the HIV persistence and the potential treatment. Thank you for your attention.